Welcome again. This episode is basically homemade. I would want you to go and take araboni, the sweet potato, and chop somewhere. I want you to make a diagram like this one. I did not get it. Diagram like this. This is araboni or a chopped potato. Make this kind of depression, and then at this point, put sugar, oblique salt. Then at this point, this you can use a sufuria. There's no problem. You're at home. At this point, use water. So you pour water and then now suspend that potato. That is an experiment that we can do at home. And then wait after some time. See what will happen. If there is a rise of solution from this point, of course, it starts to increase from this point to this point so that it's filled up, and the water content starts to go down on this part so that it's reducing in the beaker. We would say that experiment has occurred. What's happening is this. A potato chopped this way, a pawpaw, you can use a pawpaw or anything else, chopped this way, that is a cell. And I have already told you that a cell contains a vacuole. A vacuole inside it contains sugar and salt. So whenever you hear of salt and sugar from today, that is a concentrated solution. So therefore, how is this water moving from this point, from the beaker, water moving from the beaker, then to the potato, then to this point. So there are 3 points you must explain. Remember, this is a cell membrane. So therefore, inside this potato, there are cells. A cell contains sugar and salts. And that makes it, of course, hypertonic. I remember telling you that osmosis can be defined as movement of molecules from hypotonic to hypertonic. So therefore, this distilled water will move to the cells. Reason why they are moving to the cells, distilled water is hypotonic and the cells are hypertonic. Why? Because always cells have a vacuole and a vacuole contains salts and sugars. So therefore, the first process, you can't say water moved from this point to this point. No. You say 3 things. 1, water moves from the beaker into the cell by a process called osmosis due to the concentration gradient of the cells of this pawpaw. Then, diluting the cells. Once water moves inside the cells, it dilutes the cells. Once it dilutes the cells here, the cells become hypotonic to this point. So this point where you had placed sugar is now hypertonic and the cells have already been diluted. Now, the diluted cells are hypotonic and the outer side sugar solution is hypertonic. And again, there is a concentration difference, concentration gradient. Therefore, osmosis taking place, water moves from inside the cells to this point. And the process continues normally until this one rises more and the one on the beaker reduces. And that is an experiment you can always find in any paper, either in Form 1, 2, 3, 4, or in KCSE. And that is about osmosis. Very important, very short, but very disciplined explanation. See you in the next episode.